Unit 1, topics 4, 5, and 6, biological macromolecules. We're going to talk about the properties, the structures, and functions. Proteins. Now, the formation of these proteins, there's an assembly line for it, and it has different structures based on the number of proteins that are assembled. We have one monomer. We call that an amino acid. That's the smallest formation of a protein. When two amino acids join, they form a peptide. And many peptides forming, do you know how they're forming? That's right, a dehydration reaction will form a polypeptide. And then eventually these polypeptides, these long chains of amino acid, will start to turn and fold to form a larger protein. Yeah. Essentially, proteins are polymers of amino acids. They're molecules that consist of lots of polypeptides together, and this long chain of polypeptide will fold in and on itself, creating this 3D structure. And they're all comprised of carbon, hydrogen, oxygen, nitrogen, and sulfur. And whatever shape they take on will determine how that protein functions. The amino acids, when bonded together to form these polypeptides, they do form the backbone of what will ultimately be a very large protein. But what's kind of hanging off of this chain of polypeptides will form these van der Waals interactions that are hydrophobic and cause the molecule to turn in and out on itself. To understand that, we have to know what an amino acid is. So molecules, they have these amino groups and carboxyl groups. There's 20 different amino acids. But don't worry, unlike the functional groups, I'm not going to make you memorize all 20 of those because I'm not amino acid. Haha. Okay. But you do have to be able to recognize them, their general structure. Here's some examples. So an amino group and a carboxyl group, aminos NH2, a carboxyl group is the carbon double bonded to an oxygen with the hydroxyl group, carboxyl, if you think about it, hanging off of this carbon. And there's also R groups. They're what are called side chains. They vary in structure. R groups give a unique side function to amino acids. And if you look at the examples here, we have two amino acids that are very, very similar in structure, but their side chain is different. One of them is CH3, and the other one is a hydrogen. So the R group, the methyl group, is unique, and the hydrogen group give the variation in these amino acids, making one of the amino acids alanine and the other one glycine. Each amino acid has a unique side chain, and the unique aspects of the amino acid are based upon this side chain. It definitely gives it its physical properties and its chemical properties. These side chains have lots of different characteristics to them. Some of them are hydrophobic, some of them are hydrophilic, so they could definitely be nonpolar or polar. Some of them can also be charged. We call that an ionic, which is like an ionic charged hydrophilic R group. So side chains, they interact, and that basically will determine the shape of the protein and how the protein will function. So just as a thought, let's just take our hydrogen right here. Perhaps you can understand that if I have a hydrogen, which has a positive charge, hanging off of my amino acid, and then this methyl group here, which is carbon bonded to all of these hydrogens and attached to another carbon, this group right here, it's complete. It doesn't have any open charge or anything like that to it. But if I have a hydrogen right here that possesses the availability of a charge, these two things are going to interact very differently. And I'm saying this hypothetically. I'm not saying that's the case in these amino acids, but I'm just saying hypothetically, the interaction will definitely determine the shape and the function. So when these peptide bonds form, to form these peptide bonds, like the carboxyl group of one amino acid must be positioned next to the amino group of another amino acid. And through the process of a dehydration reaction, we remove water and start to create this carbon nitrogen backbone interaction. So anything that is hanging off of the repetitive structure in the center are what we call side chains, or groups. You assemble many of these together. Many amino acids will link up by these peptide bonds through a dehydration reaction, and we get polypeptides. Each polypeptide is going to have this very unique sequence of amino acids and directionality, the way that it faces, how the R groups stand off, up, bottom, left, right. You have to think about it as 3 dimension, but each end is going to be chemically unique as well. How they terminate an amino acid group, a polypeptide, is also unique and important. One end is going to have a free amino acid group. We call that the N terminus. And the other end is going to have a free carboxyl group, or we call that C terminus. If you flip back to the previous slide, one end of the polypeptide was ending in this, and then there was blah, 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 blah. There was a chain of stuff, and then it terminated in something like this. So we have C terminus and N terminus. That's what it's talking about. It's how the polypeptide orients itself in a sequence. All right, so the sequence of these amino acids is going to determine its shape. So remember, shape determines function. And when a polypeptide like twists and folds, because of those R groups that interact, we ultimately, at the end of the day, we get a protein. That's what we want. Perhaps you remember from general biology, honors biology, that these sequence and these proteins are all put together through protein synthesis, and we use our genes, our DNA, to assemble the unique sequence. Because life-functioning proteins are unique and very complex, they do form these proteins that have lots of advantages for living things. Antibodies, enzymes, anything that's a messenger, transmit signal like a hormone, anything that is structural that provides support and structure. And of course, we use them to transport things, to store things. Both animals and plant cells do. So the function of proteins include many amazing things in a variety of different functions. We'll learn a lot of them individually and more intimately, but in the gist of things, these are how proteins function differently from the other macromolecules. When proteins twist and fold on top of each other, we get a primary, secondary, tertiary, and quaternary type of folding. So the primary structure is where the linear chain of amino acids, which is again determined by our gene sequencing, it's going to dictate that chain, how it starts to twist and fold on itself. The secondary structure is when that protein chain starts to interact with the other parts of that linear piece. We get these coils and folding on themselves. And this has to do with the interaction of hydrogen bonds that are within that polypeptide backbone. In specifics, we get these beta pleated sheets, which are the hydrogen bonds between the polypeptide chains. When they start to lay on their side, they almost accordion-like. Or we can get an alpha helix type of folding, which kind of reminds us of another structure, DNA structure. And that's the hydrogen bonding between every fourth amino acid, gives it that helical effect. After we get that scrunchy type folding or twisted helical version, we have the tertiary folding, which is the 3D folding. The interactions between all of those R groups and those side chains create a stabilization in the tertiary structure. So when you superimpose the patterns of the secondary structure in a protein's tertiary structure, you get this ribbon-like model looking polypeptide. So, while the secondary structure involves the interaction between the backbone components, the tertiary structure is overall the shape of a polypeptide that's going to result from the interactions between those R groups, which are individualized, based on the amino acid. So, one type of interaction that contributes to the tertiary structure is called hydrophobic interaction. The term is a little somewhat misleading because it makes it sound like it is afraid of water, but in actuality, it's caused by the exclusion of those nonpolar substances by the water molecules. So once these nonpolar amino acid side chains are close together, van der Waals interactions take over and they help hold them together. Meanwhile, the hydrogen bonds between the polar side chains and the ionic bonds between the positive and negative charged side chains are also helping stabilize this tertiary structure. These are all pretty weak interactions in an aqueous cellular environment, but their overall cumulative effect makes the protein a unique shape and sturdy. Covalent bonds are called disulfide bridges. Many further just reinforce the overall shape of a protein. And so a disulfide bridge is from where these two cysteine monomers, which have that sulfhydryl group, the SH group on their side chains, it's when they're brought close together by the folding of a protein. The sulfur in one of those cysteine bonds to the sulfur in the other one, creating this disulfide bridge. And this is potentially best illustrated in class. But that's essentially the interaction between somebody here and somebody here, kind of creating this little interaction that holds the helical shape together. All right, when we have multiple of those tertiary folded proteins colliding together in subunits, they form a much larger protein unit, which is a quaternary structure. And that's how we really refer to proteins. They're associated by many of these polypeptides all kind of shoved together. So quaternary structure is basically overall gets the credit for protein structure. And it's just a result from all of these little tiny subunits that have interacted and come together. It's going to get names like collagen, hemoglobin, that type of thing. Often students are more familiar with the names of proteins when they're referred to in their quaternary structure. Let's practice.